Hello, everyone. Welcome to this NanoString-sponsored webinar for another LabRoots coronavirus virtual event. I'm your host. My name is Elizabeth Schneider. I'm the Senior Global Marketing Manager at NanoString. Today, we have the pleasure of presenting Dr. Camilla Margaroli from the University of Alabama at Birmingham, and she will be talking about spatial transcriptomic profiling of severe COVID-19 patients. Camilla received her PhD in Immunology and Molecular Pathogenesis in 2019 from the Graduate Division of Biological and Biomedical Sciences at Emory University. During her thesis, she studied the mechanisms leading to neutrophil plasticity in chronic diseases, with a primary focus on in infants and adults affected by cystic fibrosis. She's currently a postdoctoral trainee in Dr. Amit Gagar's group, and she continues to work on mechanisms regulating airway inflammation in acute and chronic viral infections. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by communicating with other attendees using the new live chat feature during the presentation. You can find the live chat located at the right of your screen, and you can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the abstract window below the presentation window and follow the process to obtain your credits. And thanks everyone for joining, and thank you, Camilla, for presenting. We're looking forward to your talk.
Thank you for having me, and thank you all for being here. So as Elizabeth said, today I would like to present a study that we started when actually the COVID-19 pandemic hit in 2020. So our aim was to define the transcriptional profile of the lung of COVID-19 patients to try to gain insights on this novel disease. So as you may know, COVID-19 patients and many other viral infections actually lead to ARDS, or acute respiratory distress syndrome. And this includes also the H1N1 influenza pandemic that we had previously. Both H1N1 and SARS CoV-2 actually have been characterized by increased inflammation in the lung and also increased disease-related morbidity and mortality. However, a key difference is that SARS CoV-2 patients compared to influenza patients that present with ARDS showcase an extended hospitalization, which means that probably the cellular responses in the lung are different between these two patient cohorts. So in order to address similarities and differences in these two patient cohorts, we used another novel platform offered by NanoString, which is called Spatial Transcriptomics. So it allows you to stain fixed tissue and that has been paraffin embedded, and basically label it with RNA probes that are tagged with oligos, and then also come back after that with an antibody staining as you would do immunofluorescence to identify different cells of interest. So what we did, we obtained autopsy tissue from 3 COVID-19 patients and also 3 patients that were from the H1N1 pandemic. And also we had 1 patient that was double infected with both H1N1 and COVID-19 and SARS CoV-2. So we stained these tissues for epithelium, macrophages, and also endothelium. So the 3 major cell types that you would find in the lung, and then profile the tissue both as general areas, as you can see here, so as a mixture of cells, but also from those general areas, we were able, thanks to the feature of the machine, to extract a profile just for a specific cell type. So either epithelium or endothelium, as you can see on the slide, but also just for macrophages. So after you select these areas, the machine allows you to shed the linkers and sequence the oligos from those RNAs that you hybridized, and then basically sequence those oligos and then map them back to which RNA they correspond. And we used a panel of about 1,800 genes for each area of interest or each cells of interest. So we selected initially areas that were that showcased damage or specific for the ARDS. As you can see here indicated by the black arrows, they showcase hyaline membranes, which are a key feature of ARDS. And you can see that we had them both in all three cases, both in COVID-19, in H1N1, as you can see here, but also in the double infected patient. And we extracted RNA from those, but we also went a little bit deeper in our analysis, and we decided to select areas where viral staining for SARS CoV-2 or H1N1 was highly present, as you can see in 1 and 2, although 2 is a little bit dark, H1N1 should be in red. And then areas that showcase no viral presence in these patients to see whether there was a difference. So in terms of general areas, each patient showcased a differential transcriptional profile as a total profile with SARS-CoV-2 clustering together H1N1 patient and double infected clustering in another area of the PCA analysis, suggesting that their profile is probably more similar than to SARS-CoV-2. We then proceeded to analyze the gene transcription signatures for those areas more granularly. And you can see here that SARS-CoV-2 actually had a distinct profile with several genes that were upregulated or downregulated compared to H1N1, but also compared to the double infected patient. And if we looked at the pathway analysis of these genes that were differentially expressed in these patients, you can see that there are several pathways that are actually unique to SARS-CoV-2, including innate immune system, epithelial to mesenchymal transition, platelet activation, which was already described as increased coagulation in this patient by clinical observations. And you can, as I said, extracellular matrix organization was also really increased in this COVID-19 patient, as you can see here by pathway of collagen deposition and extracellular matrix organization. As you can see, there are several genes that are implicated in that. So we next analyzed the vascular bed of the lung to see whether we could pick up the same signatures as we saw in the general areas in specific cell types. So as you may remember, we observed that there was increased genes for coagulations and also platelet aggregation. So from the general area, we selected specifically endothelial cells and the vascular beds, both in the H1N1, SARS-CoV-2, but also the double infected patient. And say the same pathway that we observed in the general tissue showed up also here, including coagulation, elevated platelet activity and aggregation, and hemostasis, so suggesting that there is a genetic or transcriptional reprogramming of these cells in SARS-CoV-2 compared to H1N1, which actually results in clinical outcomes that have been observed with increased thrombosis in this patient cohort. We then proceeded to analyze the epithelium as another major cell type in the lung. And as you can see here, there is a clear difference in genes that were upregulated in SARS-CoV-2 compared to H1N1 or compared to the double infected patient. And as of note, the double infected patients also resemble very much the H1N1 profile in all of the individual cells, but also the general areas. So as you can see here, similar to the vascular bed, we observed the same pathways that were upregulating SARS-CoV-2, including extracellular matrix organization, cell motility, integrin signaling, and so on. We then observed that COVID-19 patients actually had metaplasia of the alveoli, so the alveolar epithelium, compared to H1N1, in which it was not observed. And as you can see here, by both H&E and also the immunofluorescence staining, we observed these clusters, which were observed also in other studies. And so we profiled this cluster compared to a normal epithelium, as you saw it in the previous slide, to see whether there were any differences in terms of gene expression. So as you can see here, we phenotyped the normal epithelium compared to hyperplasia, and there were very few genes that were differentially expressed between these two epithelial states. However, in the hyperplasia, there were several genes, as you can see in the heat map, that, including KRAS, that basically are associated with cancer, but also epithelial proliferation, which were upregulated in the hyperplastic epithelium compared to the normal one in SARS CoV-2. Finally, we phenotyped also the macrophages from the tissue. And as you can see here, this is where the beauty of this technique comes handy, where you can have a mixture of cells like that, but just extract only the cells that you would love to phenotype. So this is something that is really useful, especially if you don't want to isolate them straight from the tissue, but also maintain their spatial location. So as you can see here, there were also several genes that were differentially expressed between SARS CoV-2 and H1N1, including genes that were in the basically define M1 and M2 macrophages, with SARS CoV-2 patients showcasing a very skewed M2 phenotype in these cells, as highlighted on the right in the heat map, suggesting that there are differential inflammatory processes in these two infections, although the clinical outcomes sometimes may be similar. So in conclusion, we showed you that this technique called spatial transcriptomics is actually very useful to maintain the tissue architecture. It removes the bias of isolating the cells and then perform RNA sequencing on them, while still giving a really robust transcriptional profiling of the cells of interest. There is evidence for a more fibroproliferative ARDS phenotype in SARS-CoV-2 compared to H1N1, which may implicate or may define why these patients are actually staying longer in the hospital and have a longer intubation times compared to those that are hospitalized in the ICU because of H1N1 infection. We also showed you that there is a chance for dual infection of patients with both COVID-19 and H1N1, which is probably a reality that we may look forward to in the next few years, where more and more cases of dual infection will show up. Now, we only had one patient in our cohort, so take this with a grain of salt, but if it holds true and the dual infection actually resemble more the H1N1 profile, it's actually probably a good thing, because it requires lower hospitalization times, but also a higher chance of survival. And also, there is differential immune phenotypes comparing SARS-CoV-2 infection to H1N1 or the dual infection, which should lead to probably different therapies for these two patient cohorts. However, it requires a more in-depth analysis with temporal analysis of how these transcriptional signatures develop, but also how exactly these cells interact with each other, which will require extensive studies for that. Now, for this work, I have to thank a lot of people. It was a very collaborative effort between the Department of Pathology, our the doctor that performed the autopsies, and NanoString as well, which they were really helpful to guide us through this. And with this, I would love to take any questions, and thank you for your attention.
Thanks so much, Camilla. That was a really interesting and engaging presentation. And now we will open up for our Q&A. Well, welcome everyone to the Q&A session for Camilla's talk. I hope you all are ready to ask her some questions. So please go ahead and submit them through the chat, and I will take a look now and see if there are any questions in the Q&A. So the first question that came in is actually on the next direction for your research. So Camilla, could you tell us what you might do as a next step for your research?
Sure. So I have to mention that this was not the only project going on in SARS CoV-2 in our lab, and we've been exploring more on the immune side and immune response of the lung, and basically found that there is a subpopulation of neutrophils that actually help clear the virus in the lung. So the next step will be to actually try and see the evolution of SARS CoV-2, but also what happens if there are any differences between people that develop long COVID and people that actually don't, because there aren't a lot of data out there on what could be the differences between these two subgroups of patients.
Excellent. Yeah, we look forward to seeing that data come out. So another question that came in, so this study was all about the lung, and I understand that your lab focuses a lot on the lung. Do you plan to do any of this work on any other tissues that were taken from rapid autopsies of some of these COVID-19 decedents?
Yes, so we're actually investigating the heart right now, because obviously there are a lot of complications systemically, and we found some viral staining in the heart as well, and is in concordance with data from other groups. But yeah, that's the next step. And then I know that there are other groups that are looking at the gut, for example, but we have no plans at the moment to investigate that tissue. Okay.
Yeah, I'm excited to see what your results are with the heart, because I know that there have been a lot of people that have had, issues with the cardiovascular system after having contracted SARS CoV-2. Another question that came in, did you look at any comorbidities that any of these patients had where you profiled their particular lung samples, and whether or not that, had an effect of what type of transcriptomic profiles you saw?
Yes, so that's actually a question that the reviewer also came up with. Obviously, the most severe cases of COVID will likely be linked to existing comorbidities in this population, because they are the more susceptible to develop severe COVID and then probably die of the disease. So most of our patients actually had some comorbidities. However, they were similar between the two groups, between H1N1 and then COVID. So we didn't have the numbers of patients to actually dive more into the questions of what comorbidity could lead to certain transcriptional profiles because of the limited numbers. But it's definitely an interesting question that should be answered in the future.
Excellent. Thank you so much. So let's see if we had any other questions that came in through the chat. So another question it looks like that came in is, could you comment a little bit on the GeoMx digital spatial profiling platform and how difficult or easy to use it was, potentially compared to some of the other spatial transcriptomic platforms that might be out there?
Sure. I think I may be a little biased, but I really like the platform. I If you have access to paraffin-embedded tissues, it gives you a really good flexibility on what you can study in the tissue, and also, keeping the the architecture of the tissue without isolating the cells, so that reduces the bias. The analysis part is very straightforward as well. On the platform, everything is integrated, so it's been a pretty good experience. And I think I don't know if that's known out there, but we are waiting on the single cell also transcriptional profiling to see how that will perform, because it will be a really good technique to have.
Excellent. Yeah, and I think you're referring to the NanoString spatial molecular imager that is going to be developed and released, I think, sometime next year. So yeah, we're all very anxious to see that as well. So another question that I had is, one of the advantages of using the GeoMx digital spatial profiler is the ability to select different areas of interest that you want to profile, and compared to, say, some other platforms that are out there for spatial transcriptomics that force you to kind of look at everything in the tissue, and you can't really focus in on specific architectures or tissue structures. With GeoMx, you can actually go in and almost like trace out, what parts of the tissue you want to profile. So I was wondering if you could give us an idea of like your profiling strategy of why you chose the regions of interest that you picked for your spatial transcriptomics.
Sure. So we went for the main structural cells that are resident in the lung. We picked, obviously, the epithelium, because that plays a major role in the physiology and pathophysiology. We picked the endothelium, or like vascular bed, I should say, and then the resident macrophages. However, we initially debated whether we wanted to go for the whole immune cells or just a subtype, but we decided to go just for the main structural cells that are there all the time to see their behavior in severe COVID. So we picked those cells specifically in areas of the presented lesions, so hyaline membranes, which are characteristic of ARDS that is being developed by the COVID-19 patients when they get in the severe state.
Mm-hmm. So another question that came in is, obviously a lot of these samples were taken before everyone was trying to get vaccinated. And we do know that there have been some breakthrough infections that have happened, and in some very rare cases, patients who are ending up being hospitalized after being vaccinated, in some cases twice with Moderna or Pfizer. Are you guys going to get access to any of those samples and potentially do some spatial transcriptomics on them to compare them to people that were not vaccinated?
So we haven't had many cases of people that were vaccinated and actually succumbed to the disease. So the tissues that we use,
that's what we want.
Yes, it's a really good thing. But so what we used was autopsy tissue. Obviously, you cannot really do huge biopsies of the lung when patients are still alive. It's different than with other organs, for example. So far we don't have any cases here at UAB. It's possible that probably nationally we may start to see some coming up, but I don't know whether we're going to have access to that. But it will be definitely interesting to see the main differences also between the variants too, and why certain variants are more infectious and put more people in the hospital than others.
Yeah, that's a very good point. I didn't even actually think about that. So another question that came in is, Dr. Gugger is a pulmonologist. Do you have access to any samples where potentially like a lung transplant patient, contracted COVID-19 and, looking at the spatial transcriptomics of those potential samples, and obviously there are a lot more considerations for someone who's a transplant recipient. They might be on immunosuppressives.
Yes, so there are several factors to consider when you get a transplant issue, obviously. Like they got a transplant for a certain reason, first of all, which may alter the transcriptome in the first place. And then, on top of that, you have the virus, and on top of that, you have immunosuppressants. So it will be interesting. I'm not sure whether we've had many transplant cases lately because of the shutdown of the whole medical operation because of COVID. So they limited a lot of the procedures. They're starting back up, but I'm not sure about the access to those samples, but definitely it could be possible.
Excellent. Well, thanks so much. So this is your last chance to submit questions. So we have Camilla here live, who did the spatial transcriptomic study on lung samples from severe COVID-19 cases. So please, go ahead if there was anything that you wanted to ask her. Please go ahead and submit it through the Q&A chat portion of the console. So we will give a few minutes for people to ask questions. I guess in the interim, is there anything else, Camilla, that you would like to add, to this presentation that maybe, you didn't get to talk about when, you presented it, something that's come up recently?
Not that I can think of.
Okay, excellent. All right. Well, I didn't see any more questions come through, so thanks everyone on the line for tuning in to the LabRoots Coronavirus Virtual Event Series. Really appreciated your attention. If you have any other, questions, feel free to, submit them through the chat, and if we don't get to them, LabRoots will collect them all, and I will reach out to Camilla and try to answer them as best I can. So thanks again, everyone.
Thank you.
Bye.